All right, looking good. So mollusks, this is probably I think it's the second most diverse phylum of animals. The most diverse is obviously the arthropods. The arthropods make up the vast bulk of the diversity of animal life. And but mollusks are pretty close behind. They're quite a diverse group. And it includes things like chitons and clams and snails and octopus and squid, etc. So I'd say it's a really varied group. The body designs are pretty wild. But the cool thing is the body design is kind of all based on the same essential blueprint. And that's what we're going to focus on. So the phylum Mollusca. Phylum Mollusca. All right, a couple key features that the mollusks have that I want you to know for the test, kind of some key unique things they have, is they have a shell secreting mantle. So the gland that makes the shell on these things is called a mantle. And it's really unique to this group, only they really have it. There's other animals that have shells. They don't really have a mantle like this organism has. They have a muscular foot. So their locomotion feature is essentially a muscular mucus secreting foot that they use to crawl around, kind of a unique crawling thing. Probably the closest example would be like the flatworms. How the flatworm moved around is similar, kind of by this undulating muscular motion. They have the first organism that has a dedicated respiratory structure. They have a cnidia. This could be kind of commonly called a gill, but the cnidia is the technical name for it because it's not truly a gill. But it functions like a gill. And they have a feeding radula. And the radula is kind of like a rasping tongue. The rasping tongue, like a radula, is like this rasping strap-like tongue. It's totally unique. So they have a bunch of totally unique features for the organism. The classification here, there's a couple other classes that are out there, but I'm limiting to these four because of the ones you're going to commonly see. It's not a complete list, it's good enough for our purposes. So the class Polyplecoptera, and then the class Bivalvia. Class Gastropoda. Class Cephalopoda. Okay, so that's the four we're going to do, and I'm leaving space so I can put the specimens in there and identify. They're kind of in this order as we go. Okay, so this is kind of interesting. Is the mollusk biologists, and this won't be on the exam, but there is a name for someone who studies mollusks, and it's called malacology. So malacology is the technical name for someone who's a mollusk biologist who studies mollusks specifically. You probably never heard that term before. But anyway, malacologists have come up with this kind of idea that all mollusks derive from a common ancestor. And that hypothetical common ancestor, which they call the HAM, the hypothetical ancestral mollusk, basically derived all these features. The shell, the mantle that secretes the shell, the foot, the cnidia, the radula. And so it's kind of like this sort of like archetypal mollusk. But what I want to do is I want to kind of show each mollusk and show where these features are, because essentially all the features are pretty much present. in each of these mollusks. And on the PowerPoint, I've kind of changed some terms here. The red terms are ones that I added in because sometimes, Google or whatever won't put the proper name in there. This is probably from a simpler, simplified type book or whatever. But let's cover the Polyplecoptera first. These are called the chitons. The common name for them is chitons. Anyway, the chitons probably represent the most primitive type of mollusk that we have. And what makes them primitive is, well, for one, they're all marine. There's no freshwater or terrestrial versions of these. They're all marine. And then the other thing is they have multiple shell plates. So if we look at what a chiton looks like, it is something like this. It has a body that has the mantle on top. Okay, so here's kind of the body. The head end is over here on the left, and the posterior end is over here on the right. Dorsal's up, and the ventral's down. So we have the mantle is here on top. This is the gland that kind of secretes the shell. And this one has specifically 8 shell plates. 8. So there are 8 shell plates covering the back of the chiton. And the name chiton in Greek means shield or armor. So the name chiton itself is essentially reflecting an armored body. But anyway, the 8 shell plates are on the dorsal side. They're secreted by the mantle. Okay, so that's kind of some key features there. And then the muscular foot is on the ventral side here. So this is the foot. And you can kind of see here, I've chitin flipped over, the foot is kind of like this pad that has these grooved muscular lines. And they use a combination of muscular movement, mucus, slime, and cilia to crawl along the bottom of what that's going to be. So motion feature, the foot is on the ventral side. The tenidia in these organisms is found between the foot and the mantle. The mantle actually kind of have a fold in it. There's like a little bit of a fold in the mantle. And inside that fold is the tenidia. And so you'll find them kind of along both sides. Between the foot and the mantle is a groove. And in that groove is the tenidia. When we stop looking at specimens, I'll pull some chitons out of the jar here. And on the exam, I'll have a dish with 2 chitons in it, one upright so you can see the shells, and one upside down so you can see this. And I often like to put a pin, here in the foot and ask you what's that structure. And then in this groove, the tenidia kind of looks like a rigatoni noodle, like a little rotatelli type pasta. It looks like a little curlicue, like ramen noodle or something like that in there. And that's just kind of like this increased surface area for respiration. The radula is kind of hard to see. The radula is the grasping tongue that occurs in the mouth. Let me see if I can pull up a video of this so you can kind of see what it looks like. So how about chitin radula? All kinds of fun stuff you can see here. So anyway, mute that. You can see that, let me go ahead and zoom in on this. You can see that the tongue is more than just like your tongue. It's got like stripes on it, where it's like a file. It's almost like a flexible file. On the radula, oftentimes they'll actually embed metals like magnesium and iron from the water. They absorb iron and magnesium and incorporate out of the radula. And so people who have aquariums at home, saltwater aquariums, sometimes if you get chitons in there, they'll actually scratch your glass on the inside of the aquarium. They'll actually scratch off the inner layer of glass because those radula are so hard. And the chitons are mostly herbivores, so they're mostly grazing algae off the rocks, on tide zone areas. So that's kind of like their mode of feeding. Okay, so let's see, what other features do we have? Okay, so on the exam, I'll probably have one station with a dish. I'll probably have 2 chitons in the dish, one upright so you can see the shells, one upside down. I'll ask you, what's the class name? Polyplecopora. I'll probably have a pin in, the foot, probably where the tibia is, maybe the mantle, but those are the kinds of questions that would be on the exam for the Polyplecopora or chiton. If you go out to our coast, there is actually a really cool chiton out there called the gumboot chiton. And this is this one here. You can see they're pretty big. They're like size of a football. And on those, there are 8 shells, but they're covered with the mantle. The mantle actually covers those. So here's a small one. This is a gumboot chiton. So you can see that. You can't see the shells, but you can kind of see where the shells are underneath the mantle skin on that organism. But these are really cool. Organisms. Okay. So chiton. Pretty easy. Got it? Good. All right. Bivalvia. The bivalvia includes clams, mussels, and oysters. So I'm just providing common names here. Pretty easy. That's why I'm putting them in quotes. Clams, mussels, oysters.
Yummy glass.
These are all examples of bivalvia. Kind of an interesting design. Almost all of the bivalves are filter feeders. They filter feed. So they essentially circulate water in their shell and collect microscopic food particles. So their body is kind of designed as a water pump. They're kind of pumping water through for feeding. So let me go ahead and draw a side view of a bivalve. And I always try to keep the same orientation here. The head, the anterior is pointing left, posterior to the right, dorsal is up, ventral is down. I'll do the same idea. So here is kind of a basic view of what a clam would be. Yeah, okay. And let's see, put the mouth here. Okay, so what we got going on is, again, we'll look at all the same features. We have the mantle is actually grown around the whole organism. The mantle, it's kind of like I take my chiton, and the mantle grows to cover the entire thing like a big trench coat or a big rain jacket is what's going on here. The mantle kind of comes down around and covers the whole organism. So the animal's actually living inside what's called the mantle cavity. If you were to look at this from like a head-on view, like if I were to look at the little clam, from a head-on view like this, what's going on is the mantle actually kind of goes around and covers the organism, kind of like a big umbrella or like a big coat or something. Okay, so I'll label these here. So the mantle, this is the mantle. It's really this whole thing, but really the gland that secretes the shell is limited to kind of like the very, very tips or end of the mantle. So then what happens is the shell surrounds this whole organism here, like But what's going on is, because the shell is being secreted from the mantle, you can see kind of growth rings. So one example of shells that you'll see is you'll see the shell look something like this, and then you'll see growth rings that kind of originate from the top. And the reason why the growth rings occur is because the shell is being secreted from only the outer lip of the gape or the mouth of the clam as it grows. So, so some key features: the very, very top of the shell, where the kind of the pointy point or the tippy tip of the shell on the dorsal side, this is called the umbo. The umbo is the name of the point at which the shell kind of originates. So you can kind of find the shell or look at the shell, and you can see that if you follow the growth rings back to where they start, that's called the, oh, look at that, there's a label on there from the previous test. So anyway, the umbo is the dorsal side of the clam. So the umbo kind of indicates dorsal. That's where the shell started. And then the mantle basically supplied or grew the shell kind of as it expanded out. Okay? So umbo's dorsal. The foot, in this case, the foot is this kind of little fleshy lobe here that is sticking into the shell. In bivalves, the foot is used for a variety of things. In a lot of the clams, the foot is used to burrow into the sand. So the foot kind of operates like a shovel or a hoe, where it'll pull the clam into the mud, almost like a hand or something pulling the clam in. For mussels, the foot is where the little byssal threads or the little protein fibers attach it to the rock. And so anyway, the main thing is these don't crawl. The foot's being used for burrowing or for attaching to a rock. The foot's also the tasty part of clam chowder, right? When you eat clam chowder and you eat the gummy part, that's mostly foot and mantle you're eating when you eat clams. Yum. Okay. All right. The ctenidia, on to the next thing. The ctenidia are actually like a drape or like a curtain that goes across the inside of the shell. So the ctenidia is kind of like this little curtain apparatus. If I look at the ctenidia from the side here, it's actually kind of like it kind of hangs down from the top and fills the side. So there's the ctenidia in the front view. And the ctenidia basically is like on both sides of the clam is kind of like these drapes or curtains. So the ctenidia is covered with cilia and mucus. And what happens is the ctenidia is a gill, so it's for respiration, but it's also for feeding. What happens here is these clams have typically a siphon or a some directional flow of water. So there'll be like a siphon that comes out. And we'll see this on the clams that we dissect on Thursday. But you can also see the siphon on this horse clam here. And what happens is they draw water in to the clam shell in one siphon and then expel water out the other siphon. So these siphons are actually structures that are associated with the mantle. Not all clams have obvious siphons, but siphons are where the water current is pulled in and pushed out of the clam shell. So the water is pulled in, water passes across the ctenidia. They do their gas exchange for respiration, but they also collect like microscopic algae and bacteria and organic material. And the ctenidia act like a conveyor belt, and the cilia will push the food to the mouth. And so here we have the mouth at this end of the organism. Essentially, the food is being then delivered to the mouth by the ctenidia, by filter feeding. So a good question for the bivalves, like when I have a clam on the test, I'll have a couple clams. I'll have this shell on the test for sure, asking you orientation and some key features. But I'll also have a clam dissection. Probably this clam as well will be on there for features. But the feeding is important. You should know that clams are all filtered. They filter using a media. That's not only for respiration, but now for feeding as well. Okay? Let's see, what else is going on? Oh, yeah. Okay, so behind the umbo is a elastic hinge. See if I can get the color there. There is an elastic hinge behind the umbo. This is the kind of like a rubbery or spring-like connector between the two shells. The name bivalvia obviously refers to bi. Valve refers to shells or plates. So the term valve kind of refers to the shell presence. So bivalve basically means two shells. It's actually only one shell folded in half, connected at the hinge here. So the main thing to note here, this is where I'm getting at, is the hinge is on the posterior side of the umbo, and the umbo is the anterior side. So in order to establish orientation, to know which is the head end or anterior end of the clam versus the posterior end of the clam, is you look at your clam and you go, okay, here's the umbo, and the hinge is on this side of the umbo. So this is posterior and this is anterior, right? So here's the orientation. You know, the umbo's here and the hinge is here. So this has got to be posterior and this has got to be anterior, okay? And that's important because, we also know that the umbo and hinge are dorsal. So that means that this part, which is called the gape is the opening of the shell. You know, where the shell opens, the gape is kind of like this area. That shell broke. Anyway, the gape here is called, is this, the gape is kind of where the mouth opens on the shell. But usually what happens is the gape is sealed up by the mantle. When the clam's actually feeding, the mantle is actually kind of keeping the gape sealed or closed. So when you see a shell like this with the thing fully open, the clam's actually dead. That one's not even alive. And I'll mention this on Thursday, when you go to the fish market and you buy clams, oftentimes the person behind the counter at the fish market, called a fishmonger usually, they'll grab the clam and they'll squeeze it. And if it closes and stays closed, it's fresh and they sell it to you. If it springs back open, it's dead and they'll throw it away. And that's one way you can test clams. One of the problems with clams is because they're filter feeders in the ocean, they're feeding on bacteria and small microbes. Clams can accumulate food poisoning. You've probably heard that clams are really risky for getting things like food poisoning or like things like algae poisoning. So you don't want a dead clam, you want to have fresh live clam when you go. So that's kind of the plan, the point there. Okay, so the gape is ventral. The umbo and hinge are dorsal, and the umbo is anterior and the hinge is posterior. So, glue my shell back together. You can tell which side of the shell this is using that feature. So if this is dorsal, the hinge is here, this is posterior, this is anterior, and this is ventral. That means this is the left shell, right? The left side of the plant. So the left side would be, that's what this is, this side. The right side of the clam would be this side here. So on the exam, I will have a clamshell, probably not this one because I broke it, but I'll have a clamshell and I'll ask you which side or which this is left or right on the shell. And then certainly you can see on years past I've asked what this structure is. It's an umbo and it's dorsal. This is the hinge, it's posterior to the umbo, and that's the gape, which is ventral. So orientation on the clam is really important. I think I can glue that back together. But anyway, so that clamshell is useful. We have some other ones that you can also do the orientation on. Sound good? But you can see what's cool is it's all the same parts. It's just they've modified and specialized in filter feeding. Useful. Okay. Clam diversity. Basically their habitat kind of dictates what they are. Clams typically are burrowers, like razor clams, or the clams that use for clam chowder are living in sand or mud. Mussels are usually found on rocks, and from their foot they produce these like really, really tough threads or fibers attached to the rock. So these, what are called vissel threads, locked into the rocks. So mussels are usually kind of like living in the tide zone on rocks that we're feeding. Scallops live on the bottom, and you probably have seen pictures, or I'll show a video here of scallops swimming. They can snap their shell shut, and they have these little wings here. Let's take a look at a video of scallop swimming, and you can kind of see what their habitat is. Scallop swimming. So the body design here is intended to let them do this, like they snap their gape open and close and use that like jet propulsion, and they can move around on the bottom, and then they kind of settle on the bottom. Kind of cute, huh? Anyway, so that's their habitat. But I think what I'm trying to relay is they're all basically kind of based on the same general body plan. Oysters, obviously, if you go to Humboldt, you can get oysters. They're kind of bay feeders. They attach to like logs or rocks in bays. But you probably know that pearls are actually produced by a certain special type of oyster. And what happens there is like a sand grain or an irritant will cause the organism to produce like this pearly, smooth covering. It's to kind of prevent them from being irritated or from getting an infection. But we obviously like we like pearls for jewelry, but it's actually a product of oysters. And sometimes you can find kind of irregular pearls and other kinds of bivalves. So anyway, very good. On the exam, it'll probably be this horse clam, and the labels on here are the foot is on one end. You can find the anterior side of the foot. The other end is the siphon, and in clams that burrow, the siphons can be quite long. You can have the siphons, for example, on a razor clam be 6 or 7 inches long. Okay, so I think that gets us good for the bivalve diversity for right now. And we'll have a dissection of a clam on Thursday. The gastropoda, this is among all the mollusks, the gastropods are the most diverse type of mollusk. And this includes the snails, the slugs, nudibranchs, etc. Okay, snails, slugs, and nudibranchs. So the name gastropoda means, gastro means stomach, poda means foot. So stomach foot. And the reason why they call them this is their body, which is mostly visceral mass with their guts in it, is sitting right on top of their foot. Let me go ahead and draw like a typical snail here. So hopefully you remember a little bit about snails from our lab that we had on snail classification. So here's kind of a model snail here.
Reminds me of the sheep he drew.
Okay. So there is our snail. And these have a single shell, usually covering the entire body. The shell is being secreted by the mantle not all snails have a coily spiral pattern. Remember the limpet was an example of one that didn't have a spirally pattern, or the abalone has kind of a minor spiral. The snails that actually have an enclosed shell, are only some of them. But the basic idea here is we have a shell. And so the mantle is up here on the dorsal side. The mantle has a cavity here. This structure here is called a mantle cavity. And in that mantle cavity is where the cnidia is located. So the cnidia is this, kind of a feathery looking structure in there for respiration. So they're using their cnidia just for respiration. And it's found in this cavity. So rather than being like in a groove exposed, their cnidia is kind of protected. It's like protected in this cavity. And we'll talk about what that cavity, why that's important in a second here. But it's kind of unique here. I guess it's kind of like here. I mean, the cnidia is protected here in a cavity as well. But the cnidia is in a cavity located there. Okay, and then the foot is ventral. So the foot of the snail or slug is pretty obvious. It would leave the slime trails in your garden or on your patio. The foot is the muscular foot, and then these have a radula. I forgot to mention that the bivalves don't have a radula because presumably they're feeding on microscopic organisms. They're not scraping, so they lost the radula. There's no evidence of a radula. So, but most gastropods are either herbivores, grazers. There are some predators, but basically they use their radula for feeding. Okay, radula. All right. And that's the basic body plan of gastropods. There's a really interesting gastropod called a cone snail. I think we have some shells in our collection back here. These are predators, and they use their radula like a harpoon. What the cone snails do is they'll use their little antennae, and they kind of wag them around like worms to attract fish to come in. And then when the fish gets near, they use water pressure to shoot their radula out like a harpoon and spear the fish. And then the radula has a venom in it that's like one of the most poisonous venoms known. And so if you go to the tropics, they'll often tell you never pick up a cone shell because if it harpoons you, it can paralyze you, you can drown. But anyway, they're killer cone snails. So you can take a look at that video. This is a link to a video on National Geographic. Okay, kind of a unique example of a marine gastropod is called sea slugs and nudibranchs. So we're talking about the marine versions right now. What a nudibranch is, what that means is naked gill. Nudi means naked and branch means gill or respiratory structure. These nudibranchs don't have a shell. So usually what they look like is this. They have their mouth and their feeding palps are here, and then they have their foot. But what they have basically is their cnidia are like sticking out of the back of their mantle. So the cnidia on a nudibranch are kind of exposed. They're like hanging out. You can see a wide variety of these here. You can see on this one, the cnidia is kind of like a little pom-pom. The cnidia on this one are all over the back. On this one, it's kind of like a flower. But they're called nudibranchs because there's no shell covering the organism. And I've never had a nudibranch on a test. It's called nudibranchs. It's the common name. But I have one here, so I don't know, maybe this this will be the first year I put a nudibranch on the test. It's just they're not very charismatic, you know, it looks like a little jelly ball, you know, it's kind of hard to know what it was. But anyway, that's an example of a nudibranch. They lack a shell entirely. So that gets us to, these are called sea slugs or sea hares also. That gets us to, here we have the first truly terrestrial organism. This is the first truly terrestrial animal that we're seeing in zoology. The annelids, the earthworms, they lived on land, but they're in wet soil, like in 100% humidity environments. But the gastropods have, you know, land snails and banana slugs and those types of things. These are organisms that are fully land living and not living in the soil. They're likely the last animal phylum to colonize land. I think they colonized in the Cretaceous. I'm sorry, in the not Cretaceous, in the Carboniferous. The first animals to colonize land were the arthropods. The next animals to colonize land were the amphibians, the vertebrates. And then in the Carboniferous, probably the mollusks colonized as land snails and slugs. But what I want to point out here is what a land slug or land snail is and how they've colonized land. So in a banana slug, for example, which is a really cool organism, in a banana slug, they have something going on like this. They have kind of like this saddle here. Okay, so here's kind of our banana slug here. So one thing to note about the banana slugs, this is like a terrestrial slug. Or snail. One thing to note about these is even though it looks like they don't have a shell, they actually do have a shell. It's just vestigial. So the shell is actually inside. It's a vestigial, and you know what that term means now. It's the remnants of the shell, and it's found inside their mantle, and it's about the size of a pea. It's tiny. So it's vestigial, meaning it doesn't really have any current known function, but it's still there. It's just small. Versus the nudibranchs, which have no shell at all. They're truly a non-shell gastropod. But the terrestrial slugs still have a shell. It's just not functional and that we know of, and it's really small and found inside the mantle there. But these organisms have a mantle cavity. This is kind of the main point here. They have a mantle cavity that has no cnidia. So the main thing I want to point out here is this mantle cavity is often called a lung. And it lacks cnidia. There's no cnidia at all present. And the idea here is that doing respiration on land is a different experience than doing respiration in the water. Obviously, in the water, you're not at risk of losing water. You know, in the ocean or fresh water, having water around you at all times makes it so you don't desiccate or dehydrate. But being on land, the biggest risk factor is that you're going to lose water to the atmosphere, called desiccation. So any way the organism can kind of reduce their water loss risk is possible. It is good. So the other thing is that because on land, the quantity of oxygen is so much more. There's about 35 times more oxygen on land than there is in water. And on exam 3, I'll have that as an lecture exam 3 test question. We'll go over respiration on land versus in water, you know, the respiration issues for oxygen concentration. But basically, because there's so much more oxygen on land, they don't need to have as big of a respiratory surface.
So they actually use their mantle cavity as their respiratory surface without